Good day everyone, this is Johann and welcome to my second cycle RD videos in hematology. The first thing we should do after donning the PPE is to disinfect the working area. To prepare the samples, the blood is drawn by capillary action into the capillary tube. Allow the tube to fill 3/4 full by capillary action, which are then sealed, and fill the second tube in the same manner. The sealed tubes are placed in the radial grooves of a special microhematocrit centrifuge. The sealed ends of the tubes are placed against the rubber gasket and the open ends toward the center. Balance the centrifuge by placing the tubes opposite to each other. The microhematocrit percentage is determined by placing the tubes on a special microhematocrit reader. Microhematocrit should be performed in duplicate on each specimen. The average of the two results are reported. For hemoglobin determination, fill the graduated tube with hydrochloric acid up to 2 mark on the gram scale. Read the volume using the lower meniscus. Draw the blood into the pipette up to the 20th mark. Wipe the tip of the pipette and expel the blood into the grated tube. Mix and stand for 10 minutes. After 10 minutes, add hydrochloric acid drop by drop and mix every after adding until the color matches the standard. Then record the result. For WBC counting, draw blood up to the 0.5 mark of the Toma WBC pipette. Remove the blood from the outside of the pipette with clean gauze or tissue, but be careful that the material does not withdraw any blood from the stem of the pipette. Then, add hydrochloric acid up to the 11 mark. After doing so, detach the tubing and mix in the pipette shaker for 3 minutes. Place a clean cover slip onto the new power chamber and discard the first 3 drops of the sample. Place the tip of the pipette onto the edge of the cover slip and allow the mixture to sip by capillary action. It is important that the cover slip should not be moved. Allow the mixture to be sipped on both sides. Then, once done, stand for 1 minute and read. In reading the sample under the microscope, use the LPO to focus on the lines on the 4-corner square, and then, using the HPO, count the WBCs in both chambers, add all and get the average of the counted WBCs. In WBC differential count, you must use the OIO and read in the battlement method. For platelet estimate, you may use the same slide from differential count. Count the platelets using OIO with the same pattern of WBC count. Count platelets in 10 consecutive field. Using Sysmex for automated CBC and platelet count. Click Manual and enter New Patient. Click OK 2 times and then mix the sample before aspirating. Place the tube on probe and click Aspirate button. Wait until done and then wait for the results and view it under the Explorer. For PT and APTT, we must centrifuge the samples first. Insert the disposable Sadiplast pipette gently with a twisting motion while pushing down until the pipette rests at the bottom of the vial. The blood that enters the pipette will be auto-zeroed and excess blood will flow into the seal reservoir compartment. Set a timer for 1 hour and let the pipette stand undisturbed for exactly 1 hour. Using the scale on the side of the tube, read the meniscus where the red cells are packed. The results for automated and manual CBC-PC are close to each other except for hemoglobin where there is great difference. After performing all the procedures, discard the contaminated materials to their proper waste containers. Do not forget to disinfect the working area.